again, biology learners. Welcome back to the lab. So last week, we were in the lab looking at different types of cells under a microscope. Now, why did we have to use a microscope to look at all of these different types of cells? Yeah, because they were so small. So today, the lab we are doing today, we're looking at why. Why are cells so small? So at your lab station, when you come back, you're going to see you have a graduated cylinder, you have a plastic cup, you have a beaker of vinegar, you have a spoon, a plastic knife, ruler, and then you're going to have 3 cubes, okay? Now these cubes are made out of a substance called agar. Agar is like gelatin, basically, like we make Jell-O, but I made it a little thicker so it's easier for us to handle. And inside of that, I put in an indicator called phenolphthalein. Phenolphthalein turns pink in the presence of a base. And so I also added a very strong base called sodium hydroxide in here. Now phenolphthalein turns it turns clear in the presence of an acid. Well, guess what? Vinegar, the other name of vinegar is acetic acid. So when phenolphthalein comes in contact with vinegar, you're right, it loses its pink color. So you have 3 sizes of cubes in here. We have our big cube, we have a medium-sized cube, maybe if I can get it out of the cup, medium-sized cube, and then you have a little teeny tiny little bitsy tiny cube. All right, so we're going to be doing this experiment with all 3 of these cubes. Now, when cells need to get things inside of the cell or out of the cell, how do they do that? Do they call it taxi? No. They have to move things across the membrane through a process called diffusion. Now, diffusion is just the movement of material from where there's a lot to where there's a little bit. Okay, so for example, when we put these cubes into vinegar, there's a lot of vinegar outside of the cubes, there's none inside. Vinegar is automatically going to start to move inside of the cube. Now, the reason that these cubes are different sizes is we are looking at how does the surface area to volume ratio affect how well things can move in to the cell. Okay, specifically the vinegar moving in. So when you get back to your lab, first thing you want to do is you're going to get your vinegar and you're going to measure out 100 milliliters of vinegar in your graduated cylinder. Okay? And then I'm going to pour that vinegar into my plastic cup. I'm going to use the same plastic cup and the same vinegar for all 3 of my cubes. All right, so I'm going to start off with the smallest cube here. All right, I need to take my handy dandy ruler and I need to measure basically how big my cube is, and I'm going to be measuring this in centimeters. So I measure my cube and I'm going to record that on my data table in row 1, I believe. Okay, it says size of cube. I'm going to record the size of this particular cube. All the sides should be the same, so you just need to record it once. All right, I'm going to get my timer ready, and as soon as I drop this cube into the vinegar, I'm going to start the timer. So I'm going to drop it in, start the timer. Okay, right now we have it set for 5 minutes. We may decide to change that, so make sure that you're listening to your teacher when they're giving instructions, because we may have to change how long we put it in the vinegar. Once time is up, I'm going to use my spoon again. I'm going to pull my cube out. And you will see when you pull the cube out that it's going to start to turn white, but it's going to have pink still in the middle. So you're going to take your plastic knife and you're going to cut that cube in half. And then you're going to measure, oops, I almost dropped my cube. And then you're going to measure how much of that cube is still pink. And you're going to record that on your data table. Okay? We'll be able to turn that into a percentage to see how the vinegar was able to get into the cell based on the different sizes of the different cubes. When you are finished, all of the agar cubes need to go into the trash. We are reusing the vinegar throughout the day, so the vinegar is going to get poured back into the vinegar beaker. Okay? Wash all your stuff, dry it, follow the cleanup instructions as given by your teacher. All right, guys, let's go find some things out. Have fun in lab. Make sure you are staying at your lab station. There's no need to be up wandering around. And, hey, let's turn some pink cubes clear. Have a great one.